Awesome. Thank you. Thank you so much for joining this session on the advanced filtering explained. So you might have joined previous sessions or might know our software, but I'm just going to give a little bit of an overview of Tapestri Insights, and then I'll go deep into this advanced filtering. So you know we have a graphical user interface software called Tapestri Insights, which is very easy to use, and then you can work with it to look at your variants of interest and select your clones. This is what the session last week was about. So when we train people and they want to look at the data, they often ask whether you can change parameters to modify perhaps the number of variants, the number of quality, the quality of the cells, or they might not find the variant they are looking for. So for all of that, we have these advanced filters, and we thought that these filters actually deserved a session on themselves. So that's what I'm going to be speaking about. So what you have here is just a screenshot of Tapestri Insights. There are 6 different filters, and I'm going to explain all of them. So just for you to know what you see, the histograms that you see are just the distribution of the different variants for different samples, because as you know, you can analyze more than one sample at once. So what are these filters? These filters allow you to filter variants based on these following quality metrics. So you can filter on genotype quality, you can filter on read depth, you can filter on alternative allele frequency or mutant BAF. You can filter or remove variant genotyped in X percentage of the cells. You can also remove cells with X percentage of the variants present. And you can also change the percentage of variants that are mutated in your samples. So we are going to go through them one by one. And feel free to interrupt me if there is any questions. So before I move on to explain the filters individually, I just want to highlight that these are 2 types of filters. So the 3 first ones, 1, 2, and 3, are cell filters, and they remove individual genotypes. So it is what it is depicted here in red as single squares. So look at the data. This is what a LUM file would look like. If you wish, like a matrix where all your cells are columns and your variants are rows. So if you modify any of these filters 1, 2, or 3, you will be removing individual genotypes. So these individual squares. Now, when you work with filters 4, 5, and 6, you will be removing the whole row or the whole column. And this is what you see here. You remove either the whole cell, the whole variant, or the whole cell. So let's move on to the first one. So this first filter is called genotype quality. So this filter has a range from 0 to 99. And we also have default filters. So when you work with Tapestri Insights, you don't need to worry to change any of these filters. We have a default settings, and this is what you will see below all the range that I'll show you for the filters. But some people, as I said, want to play around with them. So that's why we provide you that flexibility. So the default is 30. What it is remove with these filters? So you remove cell-specific genotypes based on genotype quality. So what is this genotype quality? This genotype quality is Phred scale confidence that the genotype assignment is correct, and that is derived from the genotype normalized likelihood of possible genotypes. So I know this might sound complex, but we will go through what this PL or likelihood of genotypes is in the next slide. So okay, let's see. So PL, as I said, is the normalized Phred scale likelihoods of the genotype considered in the variant record for each sample. So how is PL calculated? So that is based following this formula. All of this formula is coming from haplotype color and GATK. So this is not from Mission Bio, but I just thought it would be useful to give you a little bit of information on where we get this genotype quality from. So this is the formula, and without going into much details, you see there is a P here. This P value is how likely a genotype is called correctly, given the sequence data that it is observed. So to calculate the PL, we just take the log, multiply by -10, so we make it Phred scale, and then we normalize across all genotypes. So, and then we get to where the most likely genotype is 0. So to make it even easier, I'm just gonna illustrate this information with an example. So let's say we have a site. We have a site where the reference base is an A, and we have one read where the call is a T. So we also happen to have the conditional probabilities from the haplotype caller. All of this information is on the BCF file. And if you remember, and if you don't remember, all of this information is also available on our Tapestri pipeline. We provide you the BCF files as well as the FASTQ files and the BAP files. So we happen to have the conditional probabilities. So this A, as I said, there is X amount of reads and one is a T. So based on these conditional probabilities, we just follow this formula and generate the raw PL values. So log of that times minus 10. So for these A and T, there are three different genotype possible, right? You are homozygous AA, heterozygous AT, and homozygous TT. So you calculate the raw PL for these three genotypes. So minus 10 log of these conditional probabilities, you get 60, 40, and 20. So now, as you can see here, the higher the conditional probability, the lower the PL is. So because PL is the likelihood of the genotype, which means it is the probability of the genotype to be incorrect. So the lower the PL value, it means the genotype is more likely, and the higher the PL value, it is less likely. So after we have these raw PL values, we normalize. And the reason why we work with Phred score is because these values are very small, as you can see. So just taking the log and multiply by minus 10, which means their Phred score is much easier to work with 20s and 40s than with such a small number. Another good thing about logarithmic scale is when you normalize, instead of doing division or multiplication, you do subtraction or addition. So to normalize these values is as easy as making the lowest one equal to 0. So 20 minus 20 in this case, because it is logarithmic scale, becomes 0. And same with 40 minus 20 and 60 minus 20. So what do we see here? So usually you want the second lowest PL to be very different to the lowest PL, because that means that this, let's say, the heterozygous call is much, much more likely than the homozygous call. But in this case, the difference is only 20. And this is the calculation of our genotype quality. So second lowest minus lowest is 20 minus 0 equals 20, 20 GQ. If you remember, the filter I showed you, the threshold is 30. This means that this genotype calling is so bad quality that it will be filtered out. So ideally, you want a high GQ score. And you could expect that the GQ score could be even 1,000 or 2,000. Although in this case, because we work with GATK, the highest is always capped to 99. That's why our range is 0 to 99, but the threshold is 30. So in this case, this variant will be filtered out because the genotype quality of this cell is 20. Ideally, you want a high difference between the most likely genotype and the second most likely genotype, because that will give you confidence that what you are calling is likely true. So that is for the first filter, which is the genotype quality. So for all of the filters, I'm going to be telling you some exceptions on when you should change this threshold. So for this one in particular, you can think that if you go up with the genotype quality, you are more stringent. You are going to be removing variants, but you are keeping the variants that are always only present in very, very high quality cells. If you go down with your quality, you will have a lot of variants, but you likely will be introducing false positives. So this filter have really big importance when you change the percentage of mutated cells. And if you wish, we can also review it at the end of the call. But you can think, if we have a threshold of 1% mutated cells and you go down and you want to see 0.2% of mutated cells, you will likely introduce false positives there, right? So you want to be very stringent with the quality and only look at the genotypes that are in cells with high quality. So this is an exception where you could raise this value from the 30, which is the recommended or default, to even 50 or 60. So you will be only seeing variants present in cells with a genotype quality of 60 or 70. And all of that, it is based on this PL score from the BCF and haplotype caller. So if you want more information, we can provide you the links. These are all on the GATK database, and we have it on our knowledge base. And I believe we will also leave them below the video.
Emma,
yeah,
that was such a great explanation of something so complicated. I was just wondering, what goes into the quality? Is it the number of reads at that site, or the quality that's coming off of the sequencer and the FASTQ files?
Yeah, it is a lot of things. So it is the quality that comes from the sequencer. So that value is taken from the FASTQ files. It is also the number of reads that support that call. So it's not the same one read versus 9,099 to be able to make a heterozygous than 500 and 500. So that's certainly a mix between number of reads and also quality values from the sequencing.
Great, thank you.
Great. Awesome. So that was filter number one. I promise not to extend so much with the other ones. The other ones are simpler. So second one, this filter is based on read depth. So the range here is between 0 and maximum reads in log scale. Of course, it will be different depending on how deep you sequence your sample. We have also a default filter, which is 10 reads. 10 reads is the minimum that we'd recommend to be able to confidently call a genotype. So what it is removed when I change this filter, I will be removing genotypes based on read depth. So I will be removing the genotypes that are present in cells with less than 10 reads. I think that's the very generic explanation for this filter. It's really easy. I'm just going to tell you an exception on when you can go and be more relaxed with this filter. And it is if your sample happen to be under sequence. So we recommend 60 to 80x per amplicon per cell, but sometimes people just get more cells than they expected, so the sample ends up by being under sequence. And what happens if you are under sequence, you might be expecting to see a variant that is not present at first, and that could be because it is at below 10. So in that case, we will tell you to relax, so you can even, you don't need to ask even support, you can do it yourself. Relax this filter like to 9 or 8, read and see if you can recover that variant. When would you go higher? I wouldn't recommend you to go higher unless you are super over sequence, but this is not something that we will recommend. You would see everything that it is above 10 reads. Awesome. Third filter. So the third filter is the alternate allele frequency or mutant allele frequency. So what is the range for this filter? From 0 to 100. 0 is wild type, 100 is homozygous. What is the default? 20%. So we have a 20% threshold. What are we filtering out here? We are filtering out cell-specific genotypes based on mutant allele frequencies. So we are only looking at the mutant cells here. And as you can notice, there is nothing around 0. If we were looking at mutant and reference cells, we will have a huge peak around 0 because the allele frequency for a wild type or reference is 0. So in this case, we are only focusing on the mutant cells. So now, why are we, because in an ideal world, let me just go here. In an ideal world, you will expect an heterozygous cell to be 50% BAF, homozygous 100, and wild type 0. But, and this is an ideal distribution of an heterozygous mutant variant at a single cell level. So due to sampling effect, there is sometimes heterozygous that might have a lower allele frequency. And this is what happens here. So an ideal distribution of a single cell heterozygous will look like this. So you'll have a lot of cells around the 50%, but also two tails, one going above and one going below. And that is essentially based on the number of reads. So this is like if you had a bag with 50% red balls and 50% black balls. So the more balls you take from the bag, the likelier you are to get to 50%. But if you have less reads, 20 instead of 100, if you take 20 balls, you might get an heterozygous, which is not a perfect 50%, but 30%. So what I am showing you here is that even if a cell which is heterozygous has a V allele frequency of 30 and not a perfect 50, this distribution is telling us that this heterozygous is likely to be a true clone. So there is no reason to discard it. Below 20, we will be hesitant. So that's why we have our threshold in 20. Unlike on the right, what you can see is another heterozygous cell, but in this case is a false positive. Why? Do you see there is not a distribution? There is no such a nice distribution of 50 going up and going down. It is just a group of cells that are above 20. So even if this heterozygous has 30% BAF, and this heterozygous cell has also 50% BAF, the overall distribution of the sample is telling us that these two cells or these cells are likely to be false positives. So in this case, and also notice that we have removed the filter here. So you see everything from 0 to 60 in this case, just to show you that if you keep the filter here, you will even see lower quality cells.
Emma, is there a recommendation for how many cells you would want in this graph in order to feel confident in the value?
How many cells? I mean, it would all depend. It is sample by sample, because sometimes, and especially when you work with multi-sample, imagine that you are looking at a very, very important mutation, that in diagnosis you have this distribution, and then in remission you have 3 or 4 cells. So we cannot give you a threshold of minimum number of cells in order to call confidence. We would need to look at the distribution and see. So rather than telling you a hard threshold on number of cells, we would look at the distribution and how well it looks. So it doesn't matter if here we have 1,000 or 2,000 cells. This distribution is already telling us is a bad quality. So it is more the overall distribution of the V allele frequency of the cells than the number, I would say, that's important.
That makes sense. Thanks for the clarification.
And then what are the things that can lead to these false positives, right? So it could be PCR errors, for instance, as an example, even sequencing errors, and these are not unique reasons, but some reasons why we could see these false positives. Awesome. So this is filter 3. Let's just move to filter 4. So just
sorry, Gema, before we move to filter 4, we just had another question come in. Does filter 3 remove possible CMV events?
Oh, very good question. So no, because if you have a CMV event, and let's think about the two options. One is a loss of heterozygosity or loss of an allele. So if you are heterozygous and you lose one, if you lose the mutant, you become wild type. So your V allele frequency is 0. You will be part of the wild type. And remember, this filter is only for mutant. If you lose your wild type, you become homozygous. So then you will be way above the 20% threshold. You will be in 100. If there is, now let's look at the gains. If there is a gain of the mutant allele, you can increase the V allele frequency from a regular heterozygous 50%. Perhaps if you gain 1 copy, you can go up to 66 or 70 or 80%, but never below 20. So it will not remove any variants that are subject to copy number changes or loss of heterozygosity. Those will be always higher than 50% BAF. I don't know if that made sense or if you need any further clarification.
No, that was perfect. Thank you.
Awesome. Perfect. So let's just move on to filter 4. So just as a reminder, these are now global filters. We are not changing individual genotypes, but we are changing or removing the whole, in this case, whole variants. So filter number 4. What it is removed is variants genotyped in 50% of the cells. So if your variant is not present in 50% of the cells, you will not see it unless you change this, this filter. Range is 0 to 100 and the default is 50. So we usually want to see variants that are genotyped in 50% of the cells. Please note, I'm talking about genotype, not mutated. This means that the location is present and it is recognized in genotype, but not necessarily mutant. So we are essentially getting rid of low quality variants. Exceptions. When would we ask you, or when would you be interested in changing this value? So imagine that you are interested in a variant that it is located in a challenging region of the genome. And we all know examples like an NPM1, for instance. These regions of the genome are challenging to amplify, and one of the examples is due to high GC content. So if the primers fall in a high GC content region, sometimes they have a tough time to anneal, and they might not be present in 50% of the cells. So you can go lower just to look at these challenging variants. This is one of the exceptions where you would like to go lower. Imagine that you have a bulk sequencing confirmed variant in NPM1 that you don't see at first. So you can just change this filter and see if going down to 40 or 30, you see that variant. I forgot to say that the exception of filter 3, we will not actually recommend you to change this filter, the filter of the allele frequency, but I forgot to say, so I said now. Right, so that was filter 4. It's simple. If you go down, you see variants genotyped in less percentage of cells. If you go up, you want to see variants that are genotyped in 60, 70, or 80% of the cells. So now filter 5. So here we are filtering cells. We are getting rid of rows in our Loom file. Range, again, from 0 to 100, we are talking about percentage, and the default is higher than 50. So in this case, we will not recommend you to change this filter that much, to be honest, because essentially we have very complete data, and there are some filters already applied at the pipeline level. And as you can see, the distribution of our cells are very often around this range of 80 to 100%. So these are cells with X percentage of genotypes present. So how complete your cells are. We have a default on 50, but I tell you all our cells, based on the filters that are applied previously upstream on pipeline, they are very, very complete. So often you see the cells have at least 70% of the genotypes present. So in this case, we will not recommend you to change this filter. Great. So let's just move on to the last one, which is mutated variants. So unlike, I mean, in filter 3, we talked about mutant variant allele frequency, and we talked about mutated variants, right? Here we are talking about mutants as well. So the range of this filter is 0 to 100, and the default is 1%. So here you will be removing variants that are mutated in less than 1% of the cells. So what it is removed here, so you remove variants that are mutated in like 0.1, 0.2, 0.3% of the cells, essentially, and we put a threshold in 1. Why do we put a threshold in 1? So many reasons. One of the reasons is we work with GATK, and GATK introduced a lot of false positives. So actually, if you go down with these filters, you will see the number of variants increase crazily, like from 50 potential real variants to 15,000 if you kill this filter completely. That's because GATK introduced a lot of false positives. But the good thing about our data and our analysis is if you work with multi-sample, that's what we recommend if you have different time points to always load these time points at the same time, because if one variant happened to be in a 0.1% of the population of one of your samples, you will see it even though you have a filter in 1. Because the software will recognize that that's an important variant. So even if it does not meet that criteria, it will show. And I will show you in the next slide, which is the last slide. That applies to all the filters. If if you load 3 samples, one, do not make any the criteria of these filters. So if at least one, makes the criteria, so it is within threshold, you will see the rest. So here you can go low. You can go as low as you want. As I said, you can even go to 0, 1%. If you do that, I recommend you to contact support, because we have tips and tricks to get rid of these potential false positives of GATK. And one of them is actually generating a bed file or a whitelist, which is a very simple text file where you see the regions that you are interested in, and then you get rid of the rest. If you are expecting a very low frequency mutation in FLT3-ITD, or you want to see a very rare TP53 mutation and you expect it to be in 0.5 of the cells, let's just create a bed file, import it into Tapestri Insights, and you only see that region. So you get all the variants that are within these coordinates that you have imported, and then you don't get distracted with the 15 or 10,000 potential false positives from GATK. I don't know if you have additional questions regarding this mutated variant filter at this point. Okay, so if you don't have any questions on that, I'll show you the example I meant. So that's why multi-sample analysis makes the difference, and it is so important. Let's look at this NRAS pathogenic G13D variant. This NRAS, here we have 2 samples. We have a relapse and we have a remission sample. You might remember from previous sessions that you get the variant, the function, the protein, DAN score, the percentage of genotype cells, percentage of mutated, and then the information on the 2 time points. Look, please, at the remission. So at the remission time point, this NRAS pathogenic variant is genotype in 95% of the cells and mutated in 0.4. So this is rounded to 0. So 30 divided by 7,306 is 0.4. So if you loaded the remission sample alone, this mutation at first will be removed because we have a default filter at 1. But when you load it together with the relapse sample, where this NRAS mutation is mutated in 55% of the cells, it shows up without you having to change the filter. So the algorithm is so clever that it recognizes that that's an important variant for you. So of course, it does not filter it out, because that's all the magic of this high sensitivity analysis, that you load a remission sample, and even if the mutation is present in 2 or 3 cells, then you will be able to see it. That's why it is so important to do multi-sample analysis if you have the chance, but it's not necessary. It is not a must. Of course, you can analyze individual time points. In this case, just to close this discussion, if you wanted to analyze individual time points and you expected the NRAS, we will create a whitelist with those coordinates, lower the filter to 0.1 or 0.2, and it will show up.
Great. We had a question here. How many patient time points do you recommend to analyze simultaneously with Tapestri Insights?
2. I mean, as I said, ideally 2, like here, that's enough. But we have seen analysis with 3 and 4. The more, the better, just because it is also easier to identify interesting variants. As you get the percentage of mutated cells across different time points, the more time points you have, the more supporting information you have about how important that specific mutation is over time. Because you could go from 90 to 0 to remission, then to relapse to 20, and then perhaps you have a second remission. So the more, the better. But as I said, it is not a must to have multi-sample, but if you had 3 or 4, you will also get nicer fish plots.
Great.
Awesome. So that was all from me on advanced filtering. Just remind you that we have a very good knowledge base. We have actually an article on this advanced filtering. It has like 11 pages with all links to this haplotype caller and GTK and all these useful websites, as well as information on sample and general analysis. And that's the website over there. So with that,
Excellent.
Thank you very much for your attention, and let's see if you have questions.
Fantastic. Gema, this is going to be very helpful for all of our customers. So a question here, why is the default GC only 30? Shouldn't it be higher?
Well, I mean, we don't want to be so stringent with the genotype quality at first, so that's why we set up at 30. But as I said, if you want to be more stringent with the filtering, you can go up to 50. So I don't know exactly why it was specifically set at 30 as a value, but because as it is dynamic, you can easily increase if you like.
Okay. And then that was really cool to see how you could look at the variants that you care about in your last slide. And so the variant was filtered out because it was very low frequency, but what if it was filtered out because it was low quality? Could you assess sort of those other parameters or figure out why it was filtered out in the first place?
Very good question. So actually, this applies to all the filters. So if instead of being 0% mutated, happens to be in a sample with 25 genotype quality instead of 30, it will show up as well. So it will not get filtered out. How do you assess the quality? You can do it on Tapestri Insights easily. You can look at the variant and see the distribution of the quality. And that when you work with multi-sample, same, if you wanted to look with, at the remission, and in this case, the variant is not present because of a problem with quality, you could also use a bed file or a whitelist to look for that specific position, and you will see everything. So you will see very bad quality, medium quality, high quality, you will get everything. So nothing will get filtered out. And actually we will have a session, the support team, on missing variants. And that would be also really interesting because we will talk specifically about how to find variants that you are expecting and they don't show up at first. And a majority of them is the filters. You change the filters and they show up.
Great. All right, well, this was an excellent talk. We're so appreciative of the Mission Bio support team to do deep dives with us on the software and the data analysis. Thank you to everyone who joined, and we're going to take a couple weeks break from office hours, and we will be back later in June. Thank you again, Gema.
Awesome, thank you. Bye.